I'm personally becoming more and more convinced that there is one key question we should focus on at CBMM from the experimental and computational point of view. And the question is, what are the neural circuits underlying human intelligence, language, logic, and so on? Where are they and what is their motive? I think this is the key question today for the science of intelligence. Now, in general, in terms of basic proposals about neural circuits, I know two basic frameworks. One is the McCulloch-Pitts one, the proposal of specific circuits of individual neurons performing specific computation. And the recent special case of this class of neural circuits are deep nets. These are circuits with specific connectivity, with specific weights. They may be redundant, but they are wired precisely. The other one, the other broad proposal, is the neural assemblies idea, first proposed by Sherrington, more forcefully by Donald Hebb in his book Organization of Behavior, and then later by Abeles, Boutzakis, Leslie Valiant. And in this proposal, individual cells may be part of different assemblies that are created perhaps in real time when needed, and they may be involved, each individual cells, in multiple computations. And it's kind of sobering to admit that we still don't know which one is the correct theory, if any one of the two. Especially now with so many people on the bandwagon of deep learning, I think it's important to keep our mind open and therefore to speak a bit more about assemblies of neurons. And there is no better theory of neural assemblies that I know of than the recent work of Santosh, Christos Papadimitriou, and colleagues. And so I'm very happy to introduce Santosh Vempala today to speak about assemblies of neurons. Santosh got his PhD in computer science in Berkeley. He taught there and at MIT, and is now professor of computer science, actually distinguished professor of computer science at the Georgia Institute of Technology. His main work so far has been in theoretical computer science, randomized algorithms and so on, but he got in love recently with the question of how does the brain work. Welcome, Santosh.
Thank you very much. And thanks for the great introduction to assemblies and the context. I've been on this journey for the past 7 years, all of it with Christos Papadimitriou, who's been a long-time mentor and collaborator. And the question, the high-level question is roughly what Tommy just asked. How does the mind emerge from the brain? And to say it in a bit more precisely, there's fantastic, amazing, stunning progress in neuroscience and increasing insight in cognitive science. And yet an overarching theory remains elusive. How do these neurons and synapses and other molecular and chemical and electrical mechanisms result in this robust behavior that we don't understand? So that's the overarching question. And this was phrased precisely by Nobel laureate Richard Axel not that long ago, where he said, we do not have a logic for the transformation of neural activity into thought. I view discerning this logic as the most important future direction of neuroscience. So at the end, maybe if there's time, we'll discuss further motivations for this, but that's the high-level thing. And let me get straight into it. What kind of theory, formal theory, would qualify as such a logic to convert the activity of low-level hardware to cognition? And early, or now it's 25 years ago, theory was proposed by Les Valiant, the neuroidal model, where he allowed little units to change state based on what they saw from their neighbors, and thereby simulate various types of computation and memory allocation and so on. But what is it that we'd like to have in this model, in such a model for turning neural activity into cognition? Well, it should be, first of all, it should be a computational system, something that you could simulate, something that you know how states change and what the input-output behavior will be, and so on. It should be consistent with current understanding of the brain. At least it should not have some clear discrepancy. We don't need it to be precise at the level of every single reaction or chemical, but certainly we want to be consistent. And then, of course, most importantly, it should explain cognitive phenomena, some of which we don't know how to explain by any means, forget about by a neural basis. So more precisely, what would such a system consist of? What would its data types be, and what would its operations be? I mean, if we're talking about a computational system, we must have data types and operations at some level. And now, what is the right level? At least the next question. Are we talking about the whole brain, the entire brain as a network, as Camilo Golgi would have said? Are we talking about neurons and synapses, like Ramon y Cajal would have said maybe that's the seat of activity? Even lower, maybe dendrites, molecules, or is it something maybe in between the whole brain and this level of description? So this is where the assembly calculus comes in. This is our proposal, building on much work from the past. It's a formal probabilistic model of the brain. So first of all, it's going to be precise enough that you can reason about it, and a theoretician can argue what will happen, and so on. It will have a single basic data type. It'll have a handful of operations that you can do, so primitives, and it'll have a completeness theorem. So you'll be able to say that using this, you can actually simulate interesting computation. And finally, and this is certainly work in progress, we hope that it has a killer app, which hopefully is language. But yeah, this is early days for that. There's a paper that came out last year based on our rigorous formulation of these ideas. It's called Computation by Assemblies of Neurons, with Christos, Dan Mitropolsky, who's a student at Columbia, Mike Collins, a language and natural language expert at Columbia, and Wolfgang Maass, who has been doing theoretical neuroscience for decades now. So I'll start right away with the mathematical model of the brain. Okay, so this is going to be a serious abstraction, and I won't even bother to tell you about the biology. So here we go. We'll think of the brain as having a finite number of brain regions. Okay, so there are these regions, and those are finite, 10, 20, something. Each one will contain a large number of neurons, and we'll just say for simplicity they have the same number, n neurons. You could have n1, n2, and so on. And the following very important principle will be in play throughout, that in any brain area, at any given point, only k of the neurons are firing. So neurons we think of as just on or off. So they're either firing once or they're off at 0. And k, something smaller than n, significantly smaller, will be a cap on how many are firing at any given time. Okay, so there will be, and this step here, let me just remark, is inhibition. I mean, everybody might want to fire, but only the top k are allowed to fire. And then one of the beautiful things that has been well understood is the brain's mechanism for quickly regulating how many neurons are firing. So this is an abstraction of that. Now, some pairs of areas are going to be connected by a bipartite random graph. So these red arrows represent directed bipartite graph from one area to another area, with the probability of a particular edge is p, and since it's n by n, I'm calling it b and p. And every area is internally recurrently connected. So there are recurrent connections within each area. And that we'll model just as G and P. Again, the P and the P could have been different, but let's just say N neurons in every area and probability P of an edge being present. This is the base connectome, so to speak. You can already contrast this with any modern deep learning architecture. But okay, now that's the setup of the graph. Now what are the dynamics? The neurons will fire in discrete steps. So this is again a theoretical simplification. So there's this time that's going by, but in discrete steps. And what are the k that are going to fire? The k neurons that are going to fire in any one area are the ones with the highest weighted input from their neighbors, whatever their neighbors be, whether they're from the same area or other areas. You look at the way every synapse, this edge has a weight. Look at the weighted sum, and whichever k are the highest k in the entire area, they will fire, the top k. The connections between areas are allowed to be inhibited or disinhibited, meaning you can say, declare that, from A to B, it's blocked for now, or it's open again. And finally, we have plasticity, which we'll model in the simplest Hebbian version. So if there is a synapse from I to J, and I fires, and then in the next step, J fires. So I fires in step T, and J fires in step T plus 1, then the weight of this synapse goes up by a small multiplicative factor, 1 plus beta. We'll call beta the plasticity parameter. This is how much it changes if you fire in quick succession. Okay, and then in addition to this, there are global processes which are very, very simple natural things. Homeostasis is just the idea that every neuron will keep the sum of all its incoming weights held at constant or bounded, upper bounded, so that things don't explode. And forgetting refers to at some slow rate, all synapses, the weights will decay. Okay, so unless reinforced a lot in some period, things will, you'll forget, meaning you'll go back to resting state. Okay, so that's the model. Those are the two. That's the entire model. Okay, that's the entire model. There's no algorithm. It's just a model, and it describes how each part works. Now, if you want to have specific parameters in mind, we're thinking of n as several million. K, the cap within each area, is about square root of n, roughly. And the edge probability is tiny, so 1 in 1,000 or so. And this is all consistent with biology. That was the reason for these ranges. And the plasticity parameter will be about 0.1. So every time that you have this in quick succession, you get a 1.1 factor boost. So the ideas that we'll have are just this randomness. You've seen this selection, that's the inhibition, and plasticity. So that's it. And there's really only one operation underlying everything. So this is like the assembly level operation on which we'll build slightly more sophisticated operations. And that's this random, project, and cap. I'll tell you, we'll discuss that in a bit. But this refers basically to the idea that you're going to take the top k and cap. Everybody else gets set to 0. At the end, if there's time, I'll show you a quick online simulator. It's web-based, so anybody can access it. And you can run your own brain programs using these operations. Okay, so that's the model so far. How realistic is it? At one level, it's not realistic at all, but at another level, it's reasonably so. I mean, it's a formal model. It's not inconsistent with any of the biology. And given that it's an extreme simplification, maybe it's reasonably so. For example, the discrete steps assumption is certainly unrealistic. There's no reason to think that there's this coordinated activity at such a strong level. But as far as we can tell, it's likely doesn't change the phenomenon that we're able to establish. Plasticity and assemblies are used in rapid timescales. And so it is important to think about the timing. So it's our feeling, and this is just a subjective thing, that this model itself is somewhere between realism and being useful. And it's at the point where we can be rigorous and make some progress and have some insight. Okay, so let's get back to the question. What is the basic data type? I haven't defined that yet. I've only told you the model so far. And we want it to be larger than single neurons, but smaller than brain. And indeed, the basic data type will be assemblies or ensembles neurons. I should have put Sherrington on this list, as I said, but Hebb's book really brought this out. And there was work by Harris. Busaki has been working on this for a while, and his lab does both experimental and theoretical work, Yuste, and many others. Now, what is an assembly? It's going to be a large and densely interconnected set of excitatory neurons. So not the inhibitory ones that are trying to suppress other firing, but the main ones, the excitatory. These N neurons that we're talking about are all excitatory. The inhibition we've modeled away as the K-cap. And so a large and densely interconnected set of excitatory neurons in a brain area whose firing, either simultaneously or in a fixed pattern, is tantamount to our thinking of a particular thing, so a particular concept. So an assembly maps to a particular concept. A memory, a concept, a person, a name, a word, an episode, all of these will have assemblies. This is your basic memory data structure or data type. If you have an apple, you see an apple, there's somewhere in your brain, there's a subset of neurons that are mostly likely to fire when you see an apple or think of an apple or somebody says apple and so on. So that's what assemblies are. In his book, Busaki calls assemblies the alphabet of the brain. Okay, so here's the hypothesis. There is this intermediate level of brain computation. We're calling it the assembly calculus. It's implicated in higher level cognitive functions. And for this, we would need to demonstrate or review experiments, such as reasoning, planning, language, storytelling, math, music, etc. And assemblies of neurons are its basic data type. Okay, that's the first part. Now, what are the operations, right? What operations are we going to be able to do with this? First thing is going to be projection. Now remember, when I say operations, I don't want any additional control and a computer sitting on top of this doing more things. It should all work on this substrate. Association, I'll describe all these things. Pattern completion. Projection, basically, you can think of as a copy from one area to another area. So it's an assembly in one area projects to an assembly in another area. Association, two assemblies that come together. And it's something that seems to happen in cognition, human cognition all the time. Pattern completion, where you kind of have a hint of something and it completes to the full idea. It's that's ignited. You smell an apple and you think apple. Merge. This is the most complicated operation that we've managed to implement in the assembly calculus, and seems to be quite useful for language. I'll describe it later. And then reciprocal projection, where an assembly copies itself to another area, but the new copy also has a link back, so they have this tight connection with each other. And a couple of control commands. By control commands, I mean this inhibit and connections from one area to another, or disinhibit. Okay, so let's jump into it. What I'm going to do in the next part of the talk is go in some detail over two of these, the first two of these operations, projection and association. So you see a flavor of how the model works, and we'll be able to do things rigorously there. And then we'll get back to higher level questions. So for projection is something that's not just human, it's implicated in all kinds of animals. In fact, we can go all the way back to the fruit fly and its olfactory abilities, which are at some level very similar to any other animals. Odor consists of these odorants, molecules that come by. And then we have these odorant receptors, which react to particular odorants. And in the case of the fruit fly, we're talking about 50 different odorant receptors. These are like these sensory neurons that project onto these 50 neurons. And then there is this step, this strange step here, where these 50 are expanded to about 2,000 Kenyan cells. Each cell seems to be more or less a random combination, a fixed but random combination of these 50. So you've got this expansion. And after this 2,000, what goes forward up to the rest of this nervous system here is just the top 100 or so. So the top 100 are the ones that go through, the rest are silenced. So you have this. So this is the motivation for random projection in CAP. You have the sensing, after which there's this random projection to a larger space. I mean, it's still a projection, but to a larger space, maybe random lifting. And then there's a cap. You only take the top K. Okay, so that's the whole procedure. Now you could ask, okay, fine. Now, so what's happening with the random projection and cap is, if you're seeing 2,000 of these, you could think of this as sampling from the distribution, which is a weighted sum of these inputs here, right? So it's a random weighted sum of these inputs. That's what each neuron is. And you're sampling that distribution 2,000 times. So since it's a random weighted sum, it looks like a Gaussian distribution. And when we're taking the top K, what we're doing is, out of this Gaussian shower, we are taking only the tail. Those are the ones, the highest K are the ones you're keeping. So that's the operation going on. Now, going back to this, you could ask, why are you saying this is random? I mean, that's a great model, but we're talking about the fruit fly. You should be able to look at the connections. Why do you say this is a random bipartite graph? Why is this random projection? And indeed it is. So in this paper by Karen, Ruta, Abbott, and Axel, they do these measurements very precisely, and up to statistical bounds, this really looks like a random bipartite graph, except that the degree distribution is not uniform on the left hand side. So some of the neurons that come in, the 50 neurons have higher degree and some have lower degree. But conditioned on that, it looks up to low order statistics, random. Okay, so that's the process. Now, why is this any good? What are we doing here? So the main feature here is the following. Suppose we have, let's just think of this as both sides being n, okay? Doesn't really matter. And a subset or A of, let's say, k neurons fire on the left-hand side. Let me just go to it. There we go. Those are k neurons. And as a result, the top k on the right-hand side, so lots of neurons get input, but at the top k, by random projection and cap, are ignited. And now let's take another subset B, okay? Maybe corresponds to a different smell, a different combination of odor receptors, and it ignites a different subset. Now here's a question. What we'd like to have is that if A and B overlap, and the projections should also overlap. Why? Because then you'll recognize similarity as a basic step, okay? And then this suggests that if indeed that happens, maybe this is a reasonable algorithm for nearest neighbor search. Do this projection and cap, and then compare the cap, which is relatively small potentially, and is this any good at doing near neighbors? Now, it's not going to beat the state of the art, but it turns out it is actually quite good on benchmarks. And this was a paper a couple of years ago by Dasgupta, Stevens, and Navlaka. Okay, but we want to do this mathematically. So, and what with Christos, what we were able to establish is that the intersection of cap A and cap B, so A and B are both subsets of size k, and their overlap is, let's say, alpha. Right, their overlap is alpha, so alpha fraction of their k, alpha k nodes they overlap in. And you were asking, what's the fraction of overlap in the projections? Now, if it was totally random, each one was independent and random, then you would only expect a k over n fraction, because each one is picking k out of n, so the fraction of overlap should be k out of n. But in fact, it's much higher. It's k over n to the power of 1 minus alpha over 1 plus alpha. So when alpha, alpha is a constant fraction, you really get this boost in the overlap on the right hand side. In our proof, we have a denominator that's logarithmic, but still there. And as far as we can tell, shouldn't be there. So that's a question that might be nice to resolve. Okay, but going back to this, these parameters match what has been observed in fruit flies, but also locusts, and lots of insects have a similar behavior and mechanism. So why is this the case? Why is the cap higher? This is relatively easy to explain. The idea is it's probability. So you have a subset A, and you're asking. So suppose this is the cap that came out of one stimulus, and what does it mean that these x and y won? That means that what you got from the input you got from A put you over some threshold. Let's say the top k needed to be higher than some threshold. The threshold is whatever was the value of the kth highest input. So you know that the total input you got from A, which I'm thinking of as x plus y. X is the input from just A. Y is the input from the intersection, and z from here. So if there's some neuron here, it's getting input from the entire everything, some random sub input. Remember, every edge has probability p. And I've broken up the input into the x part, the y part, and the z part. And the fact that this neuron made it to the cap for A, so this belongs to cap of A, means that x plus y is bigger than t, right? It made it above some threshold. Now the question is, what is now the chance that the same neuron belongs to the cap of B? For that, you'd need y plus z to exceed t, right? Because z is new, but y is old. But the point is that this conditioning gives you an advantage. Y in general should be slightly higher than just random with respect to this threshold, and therefore you get this little advantage, and it's much higher than k over n. So that's the idea. And if you compare with simulations, in simulation with the actual overlaps that you get. This yellow line here is what the theorem proves. This purple is the conjecture. The blue is what's actually observed to be the overlap of projections, and the upper line here, intriguingly, is what we get with actual assemblies, which we'll discuss next. So keep that upper line in mind. There's something that gives you even higher overlap. Okay, so that was the fruit fly, but natural question is, does something similar happen in mammals? And actually, there's something significantly nicer that happens, which is that, well, it's based on two things. First of all, here we're just talking about one layer of projection. You're done. That's it, done, right? Bipartite graph. Now I'm talking about in mammals where you have, going back to things like the brain model I described, there's recurrent connectivity. There's connectivity between the neurons that over on the other side, and there's plasticity. Okay, so we'll use these very carefully. So here's what the toy version of the process. Here's an assembly somewhere, or a stimulus that's firing, and we look on this new area, and these are the top K, right? The top K fired. Now it happens again, but now you see in the second round, these things are firing, these K from somewhere, stimulus or assembly from outside. And in addition, the top K of the first round, this first cap C1 is also firing, right? So you've got those K and these K firing. So now the new top K could well be different. These were the ones that had the highest input from outside, but the new top K depends on who's getting the highest input when you take both of these together. And so we get a different top K, and again a different top K, right? And so on. And so the question is, you've got this process here, and there's a question: will this converge? Under what circumstances will this actually converge, right? If it's just a random graph with no plasticity, then in general it won't converge. It'll just keep on moving. But with plasticity, it's going to converge. So in fact, the theorem is that with plasticity, as long as it's above this threshold, which you can see is sub-constant, the total number of neurons that will ever be activated, looking ahead forever, as the stimulus is presented, is K plus little OK. So of course, the very first round already there's K. And the point is that the second round, it will mostly overlap with this because of plasticity, except for a few. And then even fewer and even fewer, so that the total additional thing still remains K plus little OK. Now, this doesn't mean that it's the same K that will always light up. There's going to be little fluctuations. It happens in simulation, and that's what the theory says too. But there's this, the total support is only slightly more than the K plus little OK, which means there must be roughly K neurons that are firing almost all the time for this particular. So that's the convergence theorem. I'll discuss this in a little bit more detail here. But just to show you some quick plots, each curve here is for a different plasticity. So as at plasticity 0, the total support just keeps increasing. So it doesn't actually converge. But by the time you get to plasticity, it'll just over like 0.005 for this simulation size. Eventually, you do converge. You stop activating any new neurons. You just cycle between old neurons, or just fire the same ones again and again. And once plasticity is a little bit higher, like around 0.1, and very quickly it converges. And this is another way to look at it. If you look at the number of new neurons activated, it drops very fast during this process, in about a dozen iterations on the sizes of this experiment. Okay, very quickly, the proof of convergence goes more or less like this. The first cap is the K highest ones, right? So, okay, great, out of the N. Now, the second cap receives K from this as well as from the outside still. So we can think of this as this, approximate this with this Gaussian, whose mean is now 2pk, p because of the edge probability, and the variance is 2p1 minus pk. Now there is this competition between the K previous winners. They have a light advantage, right? Because they were the ones that got the most input from the outside K. But then there's this much larger pool of N neurons. Think of K as much smaller than N. And so what we can do is we can ask for the fraction of new winners at each step. How many new winners will you get in? And that determines the threshold you must pass for your total input in order to make it to this top K. And then, so you get this threshold. Okay, great. But we want to use the thresholds to bound how many new winners. And the idea is just this. The people who won the first time, you see, because they get this boost in the plasticity from the stimulus. Stimulus was on, then this top K1, so this edge increases its weight by 1 plus beta. And so this 1 plus beta boost is enough to really give you a much higher chance of being in the second K. And of course, if you survive multiple iterations, then you get this exponential boost in your ability to survive, as meaning stay in the assembly. So that's basically the idea. One very nice aspect I'd like to point out here is the following. So you could ask, what was the advantage of recurrent connections and assemblies or just projection? Why couldn't we just stop the projection? Why did evolution give you assemblies with recurrent connections? And here's one clear advantage. Suppose instead of firing the entire stimulus, which leads to most of the assembly firing, I only fire a tiny piece of it. Okay, so somehow I activate a tiny piece of it. The point is that if you do that, and if your assembly is strong enough, then it will just quickly fill out to activate almost the entire assembly. Roughly speaking, assemblies are expanders because their connections internally are very strong compared to the outside world. So once you ignite a small constant fraction of them, it takes virtually no time to ignite the whole thing. So your memory is fully enabled. And this you can't do with just projections. Okay, so that was another one. So the next operation is association. So in association, we have two stimuli or two assemblies that are both firing and repeatedly, and together. As a result, the corresponding assemblies which had already been created come closer. Their overlap in terms of number of neurons and the strength of the connections between their corresponding neurons increases. Okay, this one, you don't have to take my word for it. It's an experiment by Eison et al. In 2016, where they were recording from human patients with electrodes directly into several hundred cells in the hippocampus. This is okay in the medial temporal lobe, and here's what they observed. So here's what they did. They showed them pictures of places that would be familiar, and then they observed which neurons are firing and how many are firing. Great. And then for different places there would be different neurons firing. And then they showed them pictures of famous people, right? And that happens, you'd have again different neurons firing. And then they did this interesting thing. They would show them together, right? A person and a place. Something that the person might never have actually seen together, but this is being done in the experiment together. And some neurons would fire. Okay, some of the ones that fired for Obama, some of the ones fired for Eiffel, some new ones, so on. And then here's what they would do. They show the picture again, right? Of say the person. And some of the neurons that were originally firing only for the place that was shown together with Obama also start firing. So literally these assemblies, if you think of the subset that's firing as an assembly, actually increased their overlap. Some neurons which were not previously in one assembly came over now after this repeated simultaneous presentation. Okay, so that's association. This is provable. This we can prove in the same model, and the proof of convergence is a little bit more complicated, but quite similar. Merge is more complicated. It has to build a backwards thing. Maybe I will skip this for now, but there's a question, we can get back to it. So to recap the assembly calculus, we have a few operations, and I'll put them all up here. Projecting from one area to another area, associating two existing assemblies, pattern completing, merge, which is about two assemblies merging into one assembly in a different area, and then some control commands which are to do with reading an area or is something firing, and then disinhibiting, meaning cut off. These connections are not going to change at the moment. And you could ask, with this setup, how powerful is this system? It turns out that you can actually perform arbitrary Turing computations. So whatever a Turing machine can do in root n space, square root n space, you can do with a constant number, 5 regions, each with n neurons. Okay, we have to make some assumptions about interference and obliviousness, yeah, sure, but nevertheless, this is already enough for simulating powerful all computations, actually, with some loss in the space usage. Okay, we're not trying to say that at all that Turing machine type of computation is what happens in the brain. It's just a pointer that we're not limiting yourself severely by sticking to these neuroplausible or motivated calculus here. Okay, so here's what I want to do in the rest of the talk. Go to learning with assemblies. How to, the random graph model is something that one could have objections with, and how to move away from this, and is there a benefit to it? And briefly, if there's time, language. I could take a question or two now, or I could proceed whichever as you prefer.
Sure. There are two quick questions. Okay. One's from Laha Ale. How can you verify the assumptions are valid? Can this model learn more efficiently than the current exists deep learning models? Does it model generalize well? Does the model generalize well?
Right. It's good. So we'll get into learning right now. So I think you'll be in a better position to appreciate that. As far as how do we know the assumptions? Yes. So the big principle throughout has been to be consistent with what is accepted in neuroscience across a series of experiments. So we've had neural recordings now for a while, and the point is that things like plasticity, inhibition, and this more recently the fact that assemblies come together and so on are all experimentally validated. And now in terms of competing with deep learning, the short answer is certainly not on benchmarks, but the hope is that there are aspects on which it will provide lessons and might find ultimately, even though it seems to be limited, suggest ways to do better learning. That's certainly the hope.
Great. And we have another one from an anonymous. Can this approach simulate, explain every neural activity as mentioned in the second slide, or only a subset of them?
Oh, certainly a subset. I mean, this is a very severely limited thing, certainly a subset. But the hope is that if you can explain an interesting subset of activity, then maybe by extending it suitably, you could explain others. Yeah, but no, absolutely, it's going to be for a small subset.
Great. And then one last quick one for now. How about garbage collection, i.e. memory reuse or memory decay? Can assemblies model those?
Yes. So this is what I meant by quickly said forgetting. So basically, just as we have, so one thing that to avoid weights exploding is normalization, homeostasis, where each neuron will sort of make sure that the sum of the weights of its incoming neurons is kept, incoming synapses is kept bounded. But also, if you do nothing, generally across the board, the weights of neurons will decay at a slow rate, not at the plasticity rate at which they increase when something positive active happens, but they will all decrease. So things that are not used will just keep decaying. And in fact, it's not just about the weights. Synapses appear and disappear continually through life. So learning, how do brains learn, right? So locally we understand it's plasticity, right? I put up only the simplest, most common rule, but there are other versions of plasticity, and it's a very interesting question about the landscape of plasticity rules, but it's local. Globally, you could ask, does the brain do gradient descent? I mean, you can do lots of great things with gradient descent. Okay, this is a topic of debate with very little convincing evidence the brain can or does do gradient descent. Okay, so in fact, this motivates a whole area of biologically plausible gradient descent alternatives. And but ultimately, we're going to stick to the question: is synaptic plasticity by itself an effective learning mechanism, right? And how effective? And then why not go further? Could it have advantages over gradient descent, right? Not only do we want to say that it can do what GD does up to some level, but can it actually have advantages? Okay, so for that, we first need to set up learning. How can the brain learn through assemblies? It's a crucial problem. And how to formulate this, I'll be up front, is unclear. How best to formulate this? We have this funny system, right? We don't have a clear input-output specification. There's some initial progress on Boolean functions, I'll mention that. But I'll mostly focus on a different approach, which is to view what's happening with assemblies, their creation and their operation as quite similar to recurrent neural networks. Okay, so that's going to be the plan. But first, let me tell you something that came out of CBMM, where Rangamani and Gandhi, and they showed how you could learn Boolean functions of two variables. Basically, what their idea was, let's have assembly areas for labels. So each label, in this case 0 and 1, will have separate assembly areas, brain areas. And then when you present, so this is a version of supervised learning, when you present an example, if its label is 0, then all connections to the one label are disinhibited. And then it's just Hebbian plasticity that changes the weights of the ones that are allowed to be happening. And then if you present the next example and that's label is 1, the connections to 0 are disinhibited and so on. So this, you do this, and it learns ands and ors of two assemblies, two inputs. They tried it on MNIST, and it works reasonably for pairwise classification, which is, people will immediately remind you, much easier than doing all 10 digits. And of course, MNIST itself is, but it's already a demonstration that plasticity can already be effective for supervised learning. Okay, so this was what they did. Now, let me go to a different view, unsupervised learning. I mean, the brain seems to be at best mildly supervised, certainly not supervised in our traditional sense of here's the label, here's the example. And also, something that's talked about a lot, it's only a few examples, relatively few examples, relatively little data. Okay, so we know that the assembly projection process preserves overlap. We can prove it in this model. But here's a question. Suppose instead of talking about single inputs, I talk about a cluster of data, a set of possible inputs which are close to each other, another set of possible inputs, right? What I'd like to do is does this process create for you an assembly, one per cluster? So it's actually generalizing. It's saying all of these guys are really one assembly. All of these guys are really one assembly. So no matter which one of them you actually see, it's the same assembly that lights up. So it doesn't matter which apple you see, you got one assembly firing. It's Fuji or Gela or whatever, Washington. And okay, so that would be nice. So we don't, so far we talked about memorization of single stimuli. Now we're talking about of distributions or of clusters. Okay, is that possible? So how do we even set this up? So here's how we set this up. Each distribution will be just a sparse Bernoulli distribution. So the input is just still 0, 1, and about k out of the n will be on. Now, the way we'll run the model is that there's this big graph. This input comes on and it projects to the big area. One example, okay, done. Plasticity happens. Next example from the same class, plasticity happens. And just 5 examples. So 5 examples come in from one distribution, one Bernoulli distribution, and plasticity happens. Next, you get 5 examples from the next distribution, the next cluster, and plasticity happens. Okay. So as usual, when I say everything operates as it should, which means that you do the random projection and top k and plasticity. That's all. No special algorithm. And here's what you get. So for example, with 4 assemblies here, these are the assemblies. This is what happens when you now look at a new stimulus from the same class. This is the first distribution, second distribution, third distribution, fourth distribution. But these are the actual assemblies, the subset of, this is just showing you the subset of neurons that are firing on presenting new stimuli from the same distribution. Okay, so the overlap within a class becomes really high, the neurons that fire. So that's why it's an assembly, 98%. And between classes is low, it's like 15%. And so this I'm showing you this with 4 clusters. If you look at a little bit more closely, what's happening in the 5 rounds, remember each example is shown only once, right? So the first time the example is shown, this is what's lighting up. Second example, this, third example, fourth example, and fifth example, and it's basically stabilized already with 5 examples. Same thing for all of these, but this distribution is different. So it's going to a different first subset, different second subset, and it stays significantly different from the other one. Okay, so that's the process. Can we prove this? Do you have any hope of proving this? Fortunately, the answer is yes, but we need to define a couple of things. So we have a notion of a stimulus class, which is just a distribution of 0, 1 to the n, but it has a core subset S of size k. And so the elements in the core subset, so you have n things, out of them there's a core subset of size k. And in here, these things are more likely to be fired. That's probability R. And then everything outside is less likely to fire. We'll call it Q times K over N. Okay. So there's a subset that's more likely to fire, meaning in the input. That's how the inputs are selected. And there's the rest of it is less likely to fire. So these are Bernoulli distributions that have these random core subsets. And so the theorem is that if we have sufficiently high plasticity, and the plasticity needs to go as the inverse square of how, the probability of the core firing. So if the core fires with smaller and smaller probability, you need higher and higher plasticity. But then you need only log n samples from each class. So that's the 5 here. And this will lead again to a stable assembly with support just k plus little o of k. So it'll quickly converge. And after that, if you give me a new stimulus from the same stimulus class, same distribution, it'll just ignite that assembly. If you make this a little bit more spread out, so in this example here, we're setting R and Q to be equal, so roughly the same number of neurons from the core and outside the core. Look at what's happening. These are the corresponding assemblies. They're quite disjoint, but these little 2 by 2 matrices are showing you the overlap of the input distribution within a class, between classes, versus the overlap of the firing neurons, the actual assembly. So you see, now once you project, it's very likely that things from the same class ignite almost entirely the same subset of neurons. That's the observation here and consistent with the theory. You can do this with multiple classes, as I just mentioned, and classification. And then, of course, once you have these assemblies that are consistently firing for something from a class, it's easy to build a classifier, just add up over those neurons, right? How that's done biologically, I don't know. That's why I'm not putting it in the model. Maybe there's a readout neuron that gets activated once the assembly is activated, so it specializes to this subset. But that's something to be worked out. Yeah, this is just showing the same thing. Now you could say, okay, great, semi-supervised or mildly supervised learning. How about a more classical problem like learning a half space? Okay, so here we have examples again from 0-1 inputs, so from the Boolean cube. And what we'll do is that we'll say a half space is those that have delta higher than the expected value in some direction v, arbitrary direction v, and then the other half space is those that have minus delta. So there's this little margin between the positive and negative examples. That's already enough to create these assemblies. This is the assembly corresponding to the positive examples. This is assembly for the negative examples. And if you look at the overlap matrix, indeed, these are just Boolean n-dimensional coordinates with about k ones. That just have a bias in one direction. The overlap is almost the same within and outside. But once you projected, created this exact same process, one presentation, 5 examples at a time, you get this much more amplified separation between these corresponding assemblies. Can this be provably learned? We have a theorem towards that. I don't think this is the last word, but it does establish something. Basically, if you have k-sparse inputs with margin 2 delta, so that's your half space separation. As long as delta is bigger than square root k, then everything from before holds. Log n presentations create stable assemblies for each half space, and the new samples are correctly mapped. Okay, so that was about learning. I'm at 4:54. I could either do one more topic or go to conclusions. Any suggestions? What is preferred?
Well, maybe conclusions, and then
questions.
I'll ask you a question about random graph models if if there are not too many other questions.
Okay, just to entice you, these slides have some very nice simulations. This was a joint work with and a beautiful process that might be very nice to understand. But this is work with Mirabel Reed. Language is very interesting to study from this point of view, and in general from the learning point of view, because it's one of the last things that the brain adapted to. In fact, it adapted to the brain's strengths. I mean, language came after human brains, and so it could be a huge invaluable lens. I mean, it's kind of this demonstration of what the brain is good at. And there's been a huge, great set of experiments. I'll just show you one, the Popel experiment from Popel's lab. He presented subjects with this series of words: friendship, hill, melt, fans, blue, guess, hits, then. Looks like complete nonsense. And then he noticed that there is this it peaks at roughly 4 hertz, which is roughly what it takes you to see these one-syllable words. Okay, this is the activity, just measuring electrical activity. And then stage 2, he reformulates them into little sentences. Bad cats eat fish, new plan gave joy, little boy kicks ball. Okay, and now he notices that there are 3 spikes. There's still one at 4 hertz, one for every word. But then there's one at 1 and there's one at 2. And if this were live, I would now ask you, what do you think the 1 and the 2 are for? And indeed, you'd see that the 2 is for phrases, bad cats, eat fish, gave joy, and so on. And the 1 is for sentences. And all of these things are happening. Great. So you're forming these little trees in your brain. And indeed, this is also experimentally validated in another set of experiments. The completion of phrases activates parts of Broca's area. And so here's an assembly calculus-based architecture for syntax in language. You see something or you get a stimulus and you look for the corresponding assembly, so kick, and it projects to this area that's going to, try to make sense of the world. And you see boy, and that projects another one, boy, great. And maybe you see ball, and these are existing assemblies that are activated. Now, kick and ball together form this verb phrase. That's this merge that I mentioned but didn't talk about. And together they form a sentence. And the sentence now can go back and make sense of the whole thing. So assemblies can be hierarchical and could potentially do parsing. Okay, I'll stop there. Now, in work that Christos and others have done, they've actually built a parser on top of these assembly calculus operations. There's very nice mathematical questions about assemblies. Most fundamental of all is, what should we define as an assembly? I mean, we have working definitions, but it's not entirely clear that's right, or the final answer. Simulator, and there are open questions on all of these aspects: the calculus itself, random graph models, sparsification, learning robustly. So one thing we've observed is that plasticity rules seem to give more robust outputs, and then of course language. Okay, thanks to all of my collaborators and to you.
Very good, Santosh. That's great. Let's go to the questions.
What about this model is novel or interesting?
Let's see. As far as I know, in terms of novelty, there isn't sort of a mathematically rigorous explanation of the creation of memories and the association of memories, even those operations, that's consistent with what we know actually can happen in the brain. That's the novelty part. Interesting. Well, the memory is perhaps the first big difference between how computers work and the brain works. Right? We don't have lookup tables or other immediate data structures. So figuring out that seems fundamental. And if we can get to how parsing might happen, that would be great. Now, the most recent experiments with language as well as with learning suggest that there's much more here at work that could really be interesting. Maybe these limitations, both in terms of what you're allowed to do to update and how you do it, could be the reason why you get this behavior that's much more robust and generalizable. So that's conjecture for now, only supported by empirical things, but you've seen what we can prove. Yeah.
Great, thanks. The next one is from CBM's Kenny Blum. It might be slightly related, but my impression is that everything up to basically the first 45 minutes of your talk is in Valiant's work, associate, merge, N, K, etc. What is different?
Okay, great. So Valiant's model was certainly inspiring and very helpful, but while those operations were around, I mean, those are operations that the actual implementation is very different. So in the neuroidal model, each neuron and each synapse has states, and it can basically decide to change state arbitrarily. Like there's a little program sitting in each neuron that can tell you how to change state, what state change to make in response to anything. So much more powerful, and one could argue, as far as we know, not plausible, at least not in that generality. But second, for example, the creation of memories, the way it works is that you actually go ahead and try to recruit new neurons every time to create an assembly. When you do an association, it's not that these things come together. You actually create a completely new thing that has common neighbors to both. So I think the implementation details are both more consistent here with biology, but also potentially allow us to do things like this unsupervised learning. So yeah.
Great, thanks. The next one's from Naveen. Most of the talk develops a model based off of neuroscience experiments, such as the random graph from fruit fly experiments. Has the inverse occurred, whereby this model predicts some new behavior, which was previously undiscovered by neuroscientists?
Yeah, that would be fantastic. I mean, these are early days, and we are working with some imaging neuroscientists to try to make finer predictions about assemblies, and especially the creation of assemblies on the fly and hierarchical creation. But it's early days to go the other direction. I mean, for example, we're pinpointing how something like merge could occur, and so it'd be great to actually find neural evidence of merge at the level of recordings from individual neurons. Yeah.
Santosh, one question from me is one assembly kind of equivalent to one neuron in the McCulloch-Pitts deep network kind of framework? And if so, you know, how many assemblies can you have per n neurons?
Right, very good. So the both excellent questions. Indeed, in terms of function, right, assemblies are doing only McCulloch-Pitts neurons can do basically anything, any circuit you can put down. Here we are saying yes. You think of a McCulloch-Pitts neuron that recognizes a particular input, yeah, or a class of inputs. That would be single. Yes, that corresponds would be a single neuron. Yes, sorry, your second question was?
Well, essentially, can I consider one assembly as one neuron with a threshold, right? Or it's more complicated, the equivalence between the two, the correspondence?
Right. I think functionally, you could say they are similar or quite close. Yes, or I should say assemblies capture many aspects of single neurons. You can build single neurons that do extremely powerful things, which I don't know how to do directly with assemblies without building on top.
Yeah.
But to answer your second question about capacity and how many you can have, you know, in principle it's n choose k, but we have to be very careful about interference. Once the overlap becomes too large, then and so at the moment it looks like quadratic in n is plausible with just these methods.
But this means if suppose I have n neurons and I can choose, you know, the kind of McCulloch-Pitts framework with precise wiring for each neuron. So each neuron is a threshold unit.
Yes.
Or I can choose to have assemblies out of this n neurons.
Then you can
can I get enough assemblies to have the same power as?
Oh no, you get even more. You get even more. I'm saying you get at the moment you can plausibly show that you can get at least quadratic in n. So if you have n neurons, you can get quadratic in n assemblies that behave reliably without interference taking over.
Yeah, because interference is the question, which is difficult. Yeah.
The next one from Sanji. How did you come up with these operators? Can a smaller set of operators also lead to Turing complete computation?
Yeah, that's a good question. This is just so each operation was consistent with some observed across the board biology, neuroscience. That's one. But second, indeed, in the small set that we do have, removing any one seems to not allow us to do the Turing complete proof. So at least it's locally minimum. Yeah.
Great.
Santosh, can I ask you now if you can to say what you left out from your talk?
Okay. Yeah. So there's a beautiful question here about convergence. So I want to just thank you, present that very quickly. And the question is just about the following process. So this is this basic convergence process that we've talked about, the top K fire, right? So, you see, the point is that the brain graph is not completely random. Of course, it does not look like GNP. We know that. So what does it actually look like? And so there's this beautiful papers that measure these, and they find that the probability of reciprocal connections, little two directed two cycles, and other small cycles is much higher. Little motifs is much higher. And so one could ask, why is this? What's the advantage of this? And so to study this, we consider the following super simple process. You have a graph, there are K vertices firing. And now you sum up the weights of edges coming in, and now the top K fires. That's it, the same process we've talked about all of that. But instead of GNP, in GNP we need plasticity. What about in other graphs? Maybe we take into account, the structure of the graph is not completely random. So a natural choice here is geometric random graphs, where you have sort of hidden variables representing your position, and the probability of an edge depends on the hidden variables. So here, for example, this is a Gaussian graph where the probability of an edge is proportional to the Gaussian function e to the minus distance squared. And as a result, so you see what happens is that this top K process converges without plasticity. Without plasticity, it still converges. And so you could ask naturally, what does it converge to? So it turns out that at least on the line, if we go to the completely continuous 0-1 interval, any decreasing function, anything where the probability of an edge decreases with distance and has so strictly decreases with distance, it will always converge to single intervals, whose size corresponds to the size of the cap, and the convergence is constant time. Okay, I won't have time to do this. When you go to higher dimension, I don't even know what the shape of the limit is going to be. If I do it in 2D, I don't even know whether what the shape is going to be. Okay, in 1D it's intervals. It's nice. So okay, that's what I was going to say. But the point is that the convergence is faster. The geometry is actually helping the convergence.
Interesting questions. Okay.
Let's say we want to compare this method with deep learning for a given task. What would it be the metric of success? With neuron assembly, it seems that not all the neurons are used when making predictions. Can we take this into account?
I see. Interesting. I mean, for a first cut, it would be nice if we just compared on the same benchmarks as you would use for deep learning.
Yeah.
Can you actually classify it to the accuracy that they can? Why not? I mean, that would be nice. But yes, short of that, we can try to measure the sparsity, if you think of energy, how many total activations in the process. But the thing that already comes up, as you saw in my last few slides, is the efficiency in terms of samples. You need very few samples here to do this. Even if you were to do a simple averaging, you need a lot of samples in a traditional supervised learning method.
And the next one's from Dimitris. How is the prediction that assemblies are almost the same for the same stimulus consistent with the fact that multiple patterns of neurons, different assemblies, give rise to the same electromagnetic field? The solution of the inverse problem is non-unique.
Right. I mean, the assemblies are at the scale of neurons, right? So the electromagnetic activity that we're measuring is a much more coarse thing. So if all your activation is, let's say, in the medial temporal lobe or one part of your visual cortex, then you can certainly have this confusion on which exactly generated it. So at this point, it's a matter of how the granularity of the measurement.
Very good. I think this was great, and we took a lot of your time, but thank you, Santosh. That's very nice work. A lot to think about. It'd be nice to come up with the critical experiments.
Yes.
To prove it or disprove it. Great. Thanks to everybody. We'll be back next week.